Welcome back to Chapter 12, Nervous System Senses. This is video 3. We're going to start off talking about the sense of smell, also known as olfaction. So, olfactory receptors are chemoreceptors because they respond to chemicals that are dissolved in liquids. Um, the senses of smell provide 70 to 80% of the sense of taste. So the reason why you can't taste when your nose is stopped up um, is because the sense of taste and the sense of smell are connected. So if your nose is blocked up for whatever reason, more than likely you won't be able to taste. This also could be an important issue, or this was an important issue when we were going through COVID, when so many people couldn't taste their food. It could have been that the COVID virus attacked the olfactory nerve. And because it attacked the olfactory nerve, people weren't able to taste. Now, I don't know if that was the case, but this is my educated theory. All right, olfactory organs. So olfactory organs, you have olfactory receptor cells. These cells, if you remember, are bipolar, meaning that there are two branches coming off of the cell body of the neurons. Um, and you have the supporting epithelial cells. Olfactory neurons have knobs at the distal ends of their dendrite covered in cilia. So remember when we talked about the terminal button or terminal bouton of the neurons. Anytime you see button or knob or something like that, think about an expanded end of a neuron. Um, Expanded end of a neuron. And in this case, you're talking about the olfactory neuron, and these particular ones have cilia on the ends of the dendrites, which is unique. We hadn't seen that yet. It covers the upper part of the nasal cavity, superior to the nasal concha, and a portion of the nasal septum. So you have olfactory receptors on each of those parts of the internal anatomy of the nose, the nasal cavity, the superior nasal concha, and part of the nasal septum. Odorants, or the things that carry smell, may bind to almost 400 different types of olfactory membrane receptors, resulting in depolarization and action potentials. So there are over 400 different types of olfactory receptors, meaning, remember, each receptor has a particular thing that it's looking for. So with 400 different types of olfactory receptors, that means that you have 400 different types of scents that you can receive information about. So this is why we're able to smell things that are very uniquely scented, and be able to distinguish the differences between different scents. Seeing, so A, you're seeing the mid-sagittal cut. So you know it's a mid-sagittal cut because you're able to see the entire right side of this face, um, where you can see the nasal cavity, you can see the different concha, so the inferior, middle, superior nasal concha, and you'll notice that the olfactory area is above that. So this, if you look closely at the image, as I zoom in, what you're seeing is these free nerve endings that are located near the base of the brain in the superior aspect of the nasal cavity. So you have the olfactory nerve, cranial nerve 1, that's coming off of the brain, sends those little projections, dendritic projections through the cribriform plate, which is part of the skull that separates the brain from the nasal cavity, into the nasal cavity so that whatever scents are inhaled and end up floating upward toward the superior nasal concha can then attach to these receptors and become interpreted by the brain. So, having a closer look, you see the ends of the receptors with the cilia on them. So, the cilia is what is located in the superior aspect of the nasal cavity, where you have the scents coming in and getting into the nose and eventually bumping into one of the dendrites so that that information can be pulled into the cell, into the olfactory nerve, and then interpreted by the brain. Once olfactory receptors are stimulated, the nerve impulses travel through openings in the cribriform plates of the ethmoid bone. So let's go back. So we already talked about this being the cribriform plate. As a reminder, the cribriform plate is a part of the ethmoid bone. And these are the openings that it's talking about. So those um, neural projections are going through those holes in the cribriform plate. The olfactory nerve goes to the olfactory bulb, leads to the olfactory tract, goes to the limbic system. Limbic system is responsible for emotions, and the olfactory cortex, so that the olfactory cortex can interpret it. So know these pathways. Olfactory nerves lead to the olfactory bulb. The olfactory bulb is what you see in cranial nerve 1. Then you have the olfactory tract. That is how the nerves send information through the bulbs and eventually go into the brain. Remember, the limbic system is what is used for emotions. You already know that, excuse me, sense can cause strong memories, cause strong responses, because smell is attached to your emotions. All right, so that is the significance of that. Um, the limbic system in the processing of sense. The olfactory bulb processes sensory impulses. The limbic system is the center for memory and emotion and provides emotional responses to certain odorant molecules. So, leading hypothesis for encoding specific smells. This is what science is currently thinking is how we're able to smell. So, each olfactory receptor cell contains only one type of membrane protein. This goes back to you remembering that each receptor in the body only looking for information on one type of substance, one type of stimulus. And in this case, that is represented by the membrane protein located on the receptor cell, so that that protein is what is, excuse me, open and ready and waiting for one of the odorant molecules to come and attach to it. Each type of membrane protein can bind several types of odorants. Usually, these odorants are similar, they are shaped similarly, which is why it can bind several types. The brain interprets the binding as an olfactory code. So, with the binding of the odorant to the protein on the receptor cell, it is basically a code, some type of code that the brain can interpret. Olfactory organs located high in the nasal cavity above the pathway of inhaled air. So, as I showed you, the cribriform plate at the superior end of the nasal cavity, that is where that is located. Olfactory receptors undergo sensory adaptation rapidly. So this is why when you walk into a room, you can smell the room almost immediately, but shortly after you get in the room, you can no longer smell the room unless you're really trying to figure out what's going on, what the smells are in the room, because your olfactory senses adapt pretty rapidly. The sense of smell drops by 50% within 1 second after stimulation. 1 second after stimulation. So, those of you who wear cologne or perfume, I recommend not putting on too much just because you can't smell it. Like after you spray it the first time, you start getting used to it. You feel like it doesn't smell as strong. Don't keep putting any on because you'll be drenching yourself while your nose has just started to adapt to it. Olfactory receptor neurons are the only ones in direct contact with the environment and can be damaged. So, all of the receptors throughout your body are being shielded from the environment in some way, shape, or form. But because those olfactory receptors are going through the cribriform plate and you actually have free nerve endings that are in the superior aspect of your nasal cavity, it is directly in contact with the external environment. It's in direct contact with the air. So if you get punched in the nose, if you inhale a very strong irritant or some kind of strong chemical, if any of those things happen, those receptors can be damaged because they're so exposed. Receptors are replaced regularly. It's very unusual for neurons to be replaced in the adult human, but your olfactory receptors, your olfactory, the receptors, yeah, the olfactory neuron receptors are replaced, in fact, regularly. So let's talk about taste. First thing is the medical term for taste is gustation or gustation. At least those are the two ways that I've heard it pronounced. Gustation is the word for the sense of taste. In order to taste, you have taste buds. These are the organs of taste. They're located on the tongue, on the bumps of the tongue. Those bumps on the tongue are called papillae. Remember, we saw this word before, papillae. This word means finger-like projections. So your taste buds are located on the finger-like projections on the tongue, they're also located on the roof of the mouth. They line the cheeks and the walls of the pharynx, which is the throat. So taste buds located on uh the papillae of the tongue, the roof of the mouth, lines the cheeks, and the walls of the pharynx. You have about 10,000 taste buds. Each of the taste buds have 50 to 150 taste cells. So each of your taste buds have 50 to 100 taste cells, meaning each taste bud has 50 to 150 little cells that are responsible for you being able to taste things and to interpret what you're tasting. Excuse me. The taste receptors are chemoreceptors. You have taste cells, taste hairs. Taste cells are modified epithelial cells that function as receptors. Modified epithelial cells that function as receptors. So each you have 100 of these modified epithelial cells, 50 to 150 of these modified epithelial cells located, associated with each taste bud. Excuse me. Taste hairs. The taste hairs are microvilli or finger-like projections that protrude from the taste cells through pores of the taste buds. These are the sensitive parts of the taste cell. So the taste hairs are the finger-like projections that come off of those cells that actually allow you to taste. Taste cells are replaced every 3 days. This is why if, say, you burn your tongue, you damage some part of your tongue, it heals relatively quickly because there's a high quick turnover for taste cells. So examples of papillae on the tongue, and you see how you have all of these little taste buds located on each of the papillae. And within each taste bud, you have, uh, excuse me, these sensory nerve fibers, you have connective tissue, you have your stratified squamous epithelium of the tongue. Um, you have the taste hairs on the end of the taste cells, etc. There are five primary taste sensations. So there are five tastes that we recognize, or at least primarily recognize: sweet, sour, salty, bitter, and umami. Sweet, sour, salty, bitter, and umami. Sweet is stimulated by carbohydrates. Remember, sugar is a type of carbohydrate. Sour is stimulated by acids. So those hydrogen ions that are prevalent in acids are what stimulate the sour taste. Salty taste is stimulated by sodium ions or potassium ions. Bitter is stimulated by certain organic compounds along with magnesium and calcium salts. So when you hear the word salt or salty, know that that salty taste is sodium and potassium, whereas bitter is tasted by different types of salts, magnesium and calcium. So make sure that if you mention, or if you write, or you have a choice of salty being the only thing that tastes salts, that is incorrect because different salts have different tastes. Umami. Umami is stimulated by some amino acids as well as MSG, which is a common flavorant or seasoning. Umami is more like meaty taste, a savory kind of meaty taste, which is why it's stimulated by amino acids, which are, of course, the building blocks of proteins, and proteins are what make up meats. Each flavor results in one primary taste sensation or a combination. Spicy foods may stimulate a class of pain receptors. So when we eat spicy foods, it may actually be the pain receptors that are being stimulated that we're associating with the spicy taste. Taste receptors also undergo rapid adaptation. So just like your smell receptors, after a certain amount of time, you can't really taste the foods anymore because your taste receptors adapt. Taste pathways. Sensory impulses from taste receptor cells travel on fibers of three different cranial nerves according to the location of the taste cell. So you remember there are 12 cranial nerves. The cranial nerves that carry taste on them are your facial nerve, cranial nerve 7, glossopharyngeal nerve, cranial nerve 9, and vagus nerve, cranial nerve 10. Yes, you do need to know these three cranial nerves and that they are associated with the sense of taste. Excuse me. You need to know the name as well as the Roman numeral because cranial nerves, if you remember, are named by their name, which typically tells you something about what it does or where it's located, and the Roman numeral, which tells you where it's located with respect to the other cranial nerves as well. The cranial nerve will conduct impulses to the medulla oblongata. Those impulses then go to the thalamus and they are interpreted by the gustatory cortex in the insula. Remember, the insula is that part of the brain that we saw that was sitting between the temporal and parietal lobes. In the picture, it pushed the temporal lobe back so you can see the insula, which is a little bit deeper into the brain matter. Excuse me. So impulses are interpreted in the gustatory cortex inside that insula. Remember, the word gustatory means to taste. So smell and taste disorders. Disorders of smell and taste can be caused by colds, flu, allergy, nasal polyps, swollen mucous membranes in the nose, head injury, toxic chemical exposure, nutritional or metabolic problems, or diseases. Oftentimes, the cause of the taste and smell disorders are not identified. So medicine in that case will end up calling it iatrogenic, meaning they don't know what caused it. Um, and then you have drugs and medications that can also affect taste and smell. So please know all the things that can potentially affect taste and smell and the causes. The sense of hearing. The ear is the organ that is used to hear. It is also the organ that is used for equilibrium. You have, or let me rephrase that. The ear contains the organ that is responsible for both hearing and equilibrium. There are three sections to the ear. You have the outer, the middle, and the inner ear, or the external, middle, and internal ear. On the outer ear or external ear, you have the auricle or pinna. This is the external part that we actually call the ear. So the ear lobe is the auricle, also called the pinna. It is the funnel shape. So our earlobe is funnel shaped so that it can collect sounds. If you were to cup your hand behind your ear and start talking or turn music on, you'll notice that your hand is helping you to collect more sounds because those sounds will end up being a little bit higher as they get into the ear. You're basically funneling them or shuffling them into the ear. This is what your earlobe does to help you hear. It actually collects and kind of guides sounds into your ear. You have the external acoustic meatus. The external acoustic meatus is an S-shaped tube. This S-shaped tube is lined with ceruminous glands, glands that produce cerumen. It carries the sound to the tympanic membrane and it terminates at the tympanic membrane. So the external acoustic meatus is the ear hole. The ear hole starts at the earlobe and ends at the tympanic membrane or the eardrum. The tympanic membrane or eardrum is just like a drum head in that it will vibrate in response to sound waves. So the parts of the external ear are the earlobe called the auricle pinna, the external acoustic meatus, which is the ear hole, and the tympanic membrane, which is the eardrum at the end or the back of the ear hole, the external acoustic meatus. The middle ear is composed of, well, the middle ear starts after you pass the tympanic membrane. So then once you pass the tympanic membrane, you have the tympanic cavity. You have the auditory ossicles, and the oval window. The tympanic cavity is the air-filled space in the temporal bone. So remember that the external acoustic meatus opens up, it leads into the temporal bone, and that is an air-filled space that we're calling the tympanic cavity. The auditory ossicles, the ossicles are little bones, three little bones, ossicles. The name of those little bones are called ossicles. These bones act like drumsticks. So they vibrate in response to the tympanic membrane vibrations, and they help to amplify the force. Amplification means that it makes it bigger. So whatever sounds are being collected by the auricle or pinna brought into the ear through the external acoustic meatus, and then vibrates the tympanic membrane. That vibration is then passed on to the auditory ossicles, and they continue to vibrate. They might make that vibration a little bit bigger so that that sound wave can continue on to the next steps, and then your brain can interpret it as sound. So the names of those three tympanic ossicles, those ear bones, are the malleus, the incus, and the stapes. Also called the hammer, anvil, and stirrup because that's sort of what they look like. So the malleus is the hammer, incus is the anvil, stapes is the stirrup. And yes, you need to know all three. All three of these, as well as all three of these, and which one of those go with which ones of those. The oval window, the oval window is also part of the middle ear. It is an opening in the wall of the tympanic cavity. The stapes, which is the third auditory ossicle, vibrates against the oval window to help move the fluids in the inner ear. So we've gone past the tympanic membrane that was the border of the external ear. Then the sound traveled through the tympanic cavity and started to vibrate the auditory ossicles. The first one was the malleus. This is the one that's closest to the tympanic membrane. The incus receives the vibrations from the malleus. The incus then sends those vibrations to the stapes, which is the bone that is closest to the oval window. As the stapes vibrates, it sends those vibrations through the oval window. And on the other side of the oval window, you have fluid in that inner ear part that becomes that takes on the vibrations that the stapes sends to it. So you have the auricle, which is the funnel that collects sounds. The auricle then leads in sounds into the external acoustic meatus. Remember, the external acoustic meatus is lined with ceruminous glands. You also see here little hairs, and the hairs, as well as the cerumen that is deposited in the external acoustic meatus, are all there to help protect the structure, to grab a hold of any dust or bacteria or little bugs that might go in there, and basically protect your ears. At the end of the external acoustic meatus, you have the eardrum or tympanic membrane, and behind that, you have the tympanic cavity. The tympanic cavity contains the auditory ossicles. You have the malleus, which is the one that's closest to the tympanic membrane, the incus, which is receiving vibrations from the malleus, and then the incus sends those vibrations to the stapes, which is in contact with the oval window, and you have fluid inside of that structure that will vibrate because of the vibrations of the stapes. Here you also notice that this talks about the nasopharynx and the auditory tube. This is the tube that leads to the throat and the nose. So the reason why is, let's say you're flying and your ears start to get clogged and you can swallow to clear your ears, or you can blow your nose to clear your ear, is because this is connected. All right. So your mouth and nose are connected to your inner ear. All right. So in the middle ear, you have a tympanic reflex. The tympanic reflex um, is described as muscle contractions that occur during loud sounds to lessen the transfer of sound vibrations to the inner ear. Loud sounds, um, muscle contractions that occur during loud sounds to lessen the transfer of sound vibrations to the inner ear and prevent damage to the hearing receptors. So, yes, you need to know the tympanic reflex. I apologize, it wasn't marked in red on this. I will correct that in the uploaded PowerPoint for that you got in class. Excuse me. The muscles that are involved in this reflex, you have the tensor tympani and the stapedius. The stapedius, you can already kind of figure out what it is, or at least what it's associated with. It's associated with the stapes ossicle. So the stapedius pulls the stapes away from the oval window. So remember, the stapes is a excuse me, sending sound vibrations to the oval window so that the fluid behind the oval window can vibrate. If you have loud sounds, then the stapedius muscle will then take the stapes bone away from the oval window so that the vibrations are not so loud or so strong, and that will help to dampen the sound that you hear. You also have the tensor tympani that pulls the malleus away from the tympanic membrane. Tympanic tympani. So when you have loud sounds, the malleus is pulled away from the tympanic membrane, so it's not receiving as much information about the sound coming from the external environment. And the stapedius moves the stapes away from the oval window so that the stapes is not sending so many vibrations to the oval window and the internal ear. So this is why when you go into a concert or a club or somewhere that has very loud music, that after a couple minutes, you're able to stand near the speaker or near the sound, and it doesn't sound quite as loud. But then you also go outside and quiet, and the sounds are muffled because these muscles pull these bones away from the various windows or membranes, so that the sound vibrations aren't as loud. So, remember, external acoustic meatus, tympanic membrane, um, the cavity, the middle ear cavity, tympanic cavity. Then you have your ear bones, so malleus, incus. Malleus is connected to the incus. The incus is connected to the stapes. The stapes is connected to the oval window. The oval window is part of the inner ear. You have the tensor tympani muscle. So this muscle is attached to the malleus so that when the sound vibrations from the tympanic membrane are too great, this muscle will contract, pulling the malleus away from the tympanic membrane so that it's not receiving as much information. And then you have the stapedius muscle, which is attached to the stapes bone. So here you have the stapedius muscle tendon. It cut the muscle away, but you see the tendon attached to the stapes so that when you have loud sounds, this contracts, pulling the stay piece away from that oval window. We are going to stop here and we'll start with the auditory tube in the next video.